Hi everyone, my name is Lauren Hoyt, and I'm very excited to share my research with you all. I'm very sorry I couldn't be there in person. I'm actually traveling with the St. Edward softball team, but I am still very excited to share it with you remotely. So, the title of my research thesis was Comparison of Italian Honeybee Immune Response in Langstroth and Top Bar Hives. So, you may know a little bit about honeybees, but you're probably not super familiar with just how much impact they have in the United States. And so, just alone in the economic aspect, honeybees actually provide a great deal of services to us. So, the first one being pollinator dependent crops. So, bees actually help pollinate many crops that we use on a day to day basis. And these crops actually equate to about $50 million in the US annually. On top of that, honeybees actually produce about $350 million worth of honey a year. And then finally, bees also provide pollination services. So you may have seen images like this bottom left one where bees are being transported around the United States to help pollinate important crops. And so these services actually equate to about $29 billion in the United States and upwards to $300 billion globally. On top of the economic impact, bees also contribute greatly to the agricultural industry. And so bees pollinate about 75% of all of the crops that are dependent on services like pollination. And so I want you to take a look at this image because this could be your breakfast with and without honeybees. And so honeybees actually help provide about 1 in every 3 bites that we consume per meal, which is a very staggering number if you really think about it. But bees are actually being impacted by a phenomenon known as colony collapse disorder. And so this is a relatively new phenomenon, and it is essentially marked by the unexplained disappearance of adult worker bees. So honeybees are essentially leaving their colonies, abandoning the queen as well as many resources, and the remaining bees that are left are actually not able to sustain the colony on their own, and so the honeybee colony actually collapses, hence the name colony collapse disorder. So this is plaguing the apiculture industry right now. And this graph actually depicts some of the losses that are reported by beekeepers beginning in 2007 and upwards to 2023. And so on the x-axis, you'll have the year, and then on the y-axis, you'll see the percentage of colonies lost. And I want to draw your attention to the blue and gray bars because the blue bar is what is considered an acceptable loss, and then the gray bar is the total annual loss of colonies per year. And so beginning in 2012, we see that the total annual loss is about double that what is considered an acceptable loss. And these numbers are only continuing to grow. We think that in 2024 to 2025, we're going to see about 60% losses in honeybee colonies. And in Texas alone, these numbers are staggering, almost upwards to 70% at the moment in 2025, and we're only in April. So I've included some headlines here just to really sink in how big of an issue this is. And these are all within the past month. And so we're seeing that this trend is only beginning to increase, and it is really impacting the beekeeping industry and potentially us if honeybee colonies continue to collapse. And so we don't know exactly what is causing colony collapse disorder, but we do have speculation that it is a multifactorial issue. So that means that multiple issues are contributing to colony collapse disorder. One of these may be malnutrition. So it may be that bees are not having enough resources to sustain their colony. Another potential factor is pathogen invasion. So there's a commonly known vector as varroa mites, which actually may introduce pathogens to different colonies, and this can be a contributing factor to CCD. Pesticide and herbicide usage in agricultural practices may also be contributing to CCD, and this is because pesticide and herbicides can cause accumulation of certain chemicals, which may actually impact the way that bees are receiving their nutrients off of certain flowering plants. Climate change as well can be a contributor to CCD. We know that drastic changes in weather patterns do affect bees. In Georgia, within the past month or so, there has been a 3-week delay in honeycomb production because of extreme weather changes. So they had a freeze followed by a drought, and this actually contributed to a 3-week delay in honey. Poor colony management by beekeepers is also another contributing factor, as well as many other environmental stressors that we may not be aware of. And so my research specifically focuses on another environmental stressor, which is the type of hive. And specifically, this is important because we know that environmental stressors can impact the way that the immune system responds, which may lead to an increased susceptibility to pathogens and other causes of CCD. And so, as I mentioned, my research is going to be focusing on two different types of hives and how that impacts the immunological capability of honeybees. And so the first hive that I'm going to discuss is the Langstroth hive. And so this hive was actually designed in the late 1800s to help maximize honeycomb production and modernize beekeeping practices. And so the way that this this hive is kind of designed is within this box, there are these removable frames. So in this left image, you can kind of see that interior. And so these frames actually have a existing foundation, plastic foundation, that kind of serves as a template for honeybees to build their honeycomb on top of. And so they're actually not having to expend as much energy because they already have this foundation to work with. And these frames are actually placed really closely together. So they're actually a lot more bees that are able to fit within this hive, maximizing the honey production. Alternatively, there is another hive known as the top bar hive, and these hives are more commonly used in Europe, but they are gaining attraction here in the United States because of their seemingly natural design. And so the way that this top bar works is that there are these wooden horizontal beams that hang within the interior of the hive. And there's actually no existing foundation. So it's just these beams, and the bees build out the honeycomb top down. So they form a free-hanging honeycomb, which is very different than that which you would see in the Langstroth hive, as the honeycomb is built outwards from the foundation. So this is considered to be a more natural type of hive because the bees are actually able to dictate the cell size of the honeycomb themselves, and they're less densely packed within this area because of that. And so there's several different advantages and disadvantages to the two different types of hives. For the Langstroth hives, the first advantage is that because they have this pre-existing foundation, it may mean that they are actually expending less energy producing honeycomb because they already have that existing template. So that may mean that they have better ability to fight off pathogens because they have greater energy reserves. They also have a greater production of honey and honeycomb, which serves as another energy reserve. However, the high density of bees may mean that the increased susceptibility to pathogens because there are so many bees within a tightly enclosed area. The top bar hive has a couple advantages as well. The first one being that it does resemble a natural hive because the bees are building it from scratch. So they're actually able to dictate their own cell size, which may mean that it's a more natural for their immunological functioning. So this natural honeycomb actually limits the amount of bees that can fit within this area, which may mean that there's a decreased susceptibility to pathogens because they're not so tightly enclosed and they won't spread those pathogens as easily. However, the disadvantages are that there is additional energy expenditure to produce honeycomb. So honeycomb is actually 20 times more energetically costly than honey to produce. And so without having that existing foundation like the Langstroth hive, they may actually be expending more energy, and they may have less energy to fight off pathogens if they were to invade. And so really the main aim of my research and question that I'm trying to get after is, are there any differences in immunological costs between these two different types of hives? And so the way I went about answering this research question was by taking bees from both the Langstroth and top bar hive and splitting them into a basal immunity group and an activated immunity group. So in the basal group, we had a group that was not fed at all. So we brought them back to the lab and immediately processed them. We also had another group that we fed a 1 microliter nectar solution. And so this nectar solution is essentially just water and sugar. And so this is what they would forage on in the wild. We then also took bees from the Langstroth and top bar hives and split them into an activated immunity group where we fed them an LPS solution. So LPS is stands for lipopolysaccharide, and it is a molecule found on bacteria. And this kind of serves as a way for the immune system in the bees to recognize a particle as non-self. And so this LPS molecule is targeted by the bee's immune system, and they are able to initiate an immune response in response to that LPS. And so after feeding them the LPS, we waited either 0, 1, or 2 hours to process the bees. And so as I just mentioned, kind of how we did this, just so you can get a more visual understanding, is we took bees from the colonies that we actually have here on St. Edward's campus. We then brought them back to the lab and either fed them or we didn't feed them according to the procedure I just mentioned. So this is a video just kind of showing you exactly how we fed them. So we used micropipettes and actually fed them the sugar solution or the LPS solution. And we actually made sure that they ingested all of it. Otherwise, the bees were discarded and the data was ignored. We then went ahead and processed the bees. So by that, I mean we dissected off the bee's thorax, which houses the hemolymph, which is where most of the immune response is going to occur. From there, we went ahead and grinded the thorax up and placed it into a buffer solution. We then centrifuged and pulled off the supernatant after homogenizing the sample, and this created a pure sample of hemolymph that we were able to run some assays on. And so the assays that we chose to run were a Red 660 assay, and this tells us our total protein concentration that we found within the sample. We also ran a prophenol oxidase assay, otherwise known as a PPO assay. And so this assay actually shows us kind of the immunological activity of the honeybees. And the way that this works is prophenol oxidase is actually a precursor molecule to a highly conserved pathway within invertebrates known as the encapsulation pathway. So as you can see in this image, these are a bunch of different immune responses that are conserved among invertebrate species, but the encapsulation pathway uses melanin to engulf a foreign particle, and PPO is a precursor molecule to that. So by testing the amount of PPO, we are actually getting a better understanding of immunological functioning within the honeybees. And so here are our results that we found. And just to orient you on the graph, so on the x-axis, we have our basal and activated immunity groups. And then the white bars represent our Langstroth top bar is in gray. And so the letters above the bars actually denote whether or not they share significance. So if they share the same letter, they are not significant from one another. But if they have different letters, they are significant from one another. On the y-axis, we have our prophenol oxidase activity. So this is really just the change in optical density over the total protein concentration. And so the first thing I want to draw your attention to is this basal immunity group, and specifically the large difference between the basal and the basal nectar group. So just a reminder, our basal group, we did not feed all. So we brought them back to the lab and immediately processed them, their hemolymph samples. However, the basal nectar group, we were actually able to feed that nectar solution. And so we see a really staggering difference between these two. And one of our hypotheses is that we think that because these bees were actually fed, we stressed them out so much that they weren't able to elicit a proper immune response. Nextly, I want to draw your attention to is the Langstroth responses. And so specifically the Langstroth bar at 0 minutes. So at 0 minutes, we see a really aggressive immune response early on. But I want to draw your attention to 0 because 0 in our experiment is not really 0 minutes. Because of our protocol and experimental design, processing the bees actually takes about 5 minutes. So all of these samples are actually about 5 minutes after the time that it says on the y-axis. And so this is important because at 0 minutes, we're actually looking at about a 5-minute immune response. And so we can see that within 5 minutes of receiving a treatment or that LPS solution, bees within the Langstroth hive are eliciting a very strong immune response very early. And then we see at 60 minutes and 120 minutes, which are really about 65 and 125 minutes, this starts to degrade a little bit. And we think that's either because they've either run out of energy reserves needed to continue fighting off the pathogen, or that the pathogen has been completely removed. And so we did not see that similar trend in the top bar bees. We actually saw no significant difference among the basal and activated immunity groups. And so just to conclude and kind of summarize what I've just shown you in this graph, so we found that feeding bees actually had a very significant immunological cost among those immunity groups, the basal immunity groups. And so we saw in that left side of the graph, those two staggering differences between the basal and basal nectar. And then additionally, we saw that Langstroth bees had a significantly different immune response at 0 minutes. And again, we think that is because they are able to elicit a very fast immune response early on. And this may be, as we hypothesized, that because they have that existing foundation, they're not expending so much energy on other aspects or maybe other immunological threats. So they're able to better combat our LPS solution or our simulated pathogen, if you will. And lastly, we didn't see any significant difference in immune response among the top bar bees over time, which we think may mean that they just simply don't have the energy reserves in place because they're spending so much energy creating honeycomb. And so with that, I would just like to thank Dr. Stephenson, who is my mentor, and he has been such a great help to me in conducting this research, and I really appreciate his time and his effort. I'd also like to thank those who contributed to the funding of this research, so the Brother Omard and Gold Research Funds, as well as the Supporting Ecology and Data Science Scholars NSF grant. And with that, I'm sorry I can't answer any questions in person, but if you do have questions, I'd be more than happy to answer them. So I've included my email up here. But I thank you all for your time, and I really appreciate being able to present. So thank you guys so much. And yeah, if you have any questions, please reach out.